Hello, everybody, and welcome to the Data Science Zoominar. My name is Erica Fike. I'm the program manager for the department. We are going to give people another minute or so to join, and then we will begin.
Thank you, Erica, and welcome everyone to this Zoominar with Cristian Tomasetti. Cristian is an associate professor of oncology at Johns Hopkins and one of the leaders of the application of mathematical modeling to cancer evolution, and as well as someone who is now branching into a variety of new and important topics associated with the interface between mathematical modeling, artificial intelligence, machine learning, and cancer prevention. Cristian was at Dana-Farber for a little while, working with myself and others. And I want to start out just by admitting flat out that we miss you. We really, really miss you, Cristian. It's been almost 8 years since the famous July 1st of 2013, when you drove your wonderful family down from Boston to Baltimore to start your career at Hopkins. And since then, you've really established yourself as a paradigm shifting investigators. The oncology department at Hopkins is the oldest such, and it's been characterized as always as being the house of really visionary individuals who have been able to turn the field around in unexpected and fundamental ways, and you've been no exception. You fit right into that mold of great innovators. And so the way I'm thinking we might use the time today is by trying to give the folks on the Zoominar a sense for how that journey has been for you, how the paradigms that you became familiar as a student and a postdoc have, how they struck you as not being adequate or relevant for understanding the important problems in cancer research, and how you've moved towards rocking them, changing them, twisting them, flipping them, making them even more controversial. So without much further ado, if that's a plan that works for you, I'd like to start exactly by that, by asking you what was the standard paradigm in cancer etiology when you started your career, and how you use mathematical modeling to begin challenging it.
Well, let me first thank you for your extremely kind introduction, Giovanni. That trip that you mentioned started with a baby delivered on the way to Baltimore. So that's how my adventure started. I have some slides. Is it okay if I put on the screen and show some pictures? It may help.
Absolutely.
Okay. So let's see. Let me start here. Yeah, so this is how it started. First of all, as you asked, I'll describe what the paradigm was, and in a sense still is in many ways. So here are some screenshots of how the causes of cancer were described by several institutions, and as well as Wikipedia. And so this was across the board, from organizations that were directed to the general public, all the way to the more scientific websites. And here is another example. This is by IRC, which is essentially the cancer research branch of WHO. And again, for every organ, you see long lists of environmental factors and some inherited factors. And this was, what you will find if you Google what causes cancer. Here is another example, the American Cancer Society in 2014, where, just to be brief, but essentially the number that was given was that about 75 to 80% of cancers could be associated to the environment and the rest to inherited factors. Here is another one, Harvard Epidemiology. This is a consensus statement where there was this table. I had to use Wayback Machine actually to find it's gone now. But where he stated that nearly 2/3 of cancer deaths in the United States could be linked to smoking, obesity, and lack of exercise. And if you looked at that table at that time, essentially it was the sum of the first three things in that list added to about 2/3, 65% or so. And and this interestingly, the list, the sum of it was 100%. And so I think it's fair to claim, that the description really across the board, and really the paradigm, was that cancer was by and large preventable, and that it was due to many harmful environmental and lifestyle factors, as well as some inherited factors. And at the same time, there was this, for example, person, Michael Bishop, who stated in the documentary about the Emperor of All Maladies, beautiful documentary, I suggest if someone hasn't seen it yet. He states essentially that we don't know the causes for the majority of the cancers. And similarly, we had estimates that seem to depict a slightly different picture from what the paradigm seemed to be. And so something was, I think major was missing. And so given our understanding of cancer, and of course it's a very complex disease, as we all know, and but there is this major, I call it engine, which is provided by the driver mutations. My interest was about understanding what just the basic normal background mutation rate could cause. I think the main reason was that if I'm trying to understand how much of cancer, say, can be attributed to an environment or to inherited factors, I first of all need to know what the noise is, the background noise, to try to detect the signal. And so it started all actually still in Boston with you, as you know very well. In fact, I think at that time, even after publishing it, I don't think we realized the full consequences of this paper. And because really the answer was already in many ways here, at least the first major answer. And so I won't describe the model here. I'll describe something maybe later of some modeling, but here is where I think for the first time, just using relatively simple modeling tools, mathematical tools, we try to estimate how many mutations would you expect at the end of the development phase in a tissue, and during homeostasis, depending on the age of the individual, and then once you have the full-blown cancer. And the answer, if you just develop the model and plug some estimates from the literature, was quite surprising, because it seemed to say that in fact a lot of the mutations would have been there no matter what. And that was very much against the idea at that time. If you remember, even, and I mentioned this to Bert a few times, but even Bert at the beginning was kind of surprised by this. And so that was what I think now really an important insight, as well as, this allowed us to simple regression, essentially, robust regression, to estimate the slopes of this accumulation of mutations. There was with cancer data, so that was if you want the limit. But on the other side, if you control for stage and for cancer type, the difference should have been really given by what happened before the cancer. And so I think we can claim that these were the first in vivo, exome-wide mutation rates, tissue specific. And what was surprising also there was that the estimates given by this simple regression were in the same ballpark that what was estimated to be, through other type of experiments, cell lines and so on. And so at least hinting to the fact that maybe this background mutation rate did have some major role in in the accumulations of the mutations that heal the cancer. And so here I have a picture, if the pre-2013 idea was essentially the normal accumulation of mutations before the clonal expansions count almost nothing. And I remember asking some doctors some numbers, and really was, the guess was maybe 1% or less. I think that paper provided, opened a window to the fact that actually it could have been even the majority of it. And so, in this way. And so that really, once I moved to Baltimore, this was in my mind, and I started thinking, how can we understand in other ways what the role of what I call, bad luck, or R for replicative mutations could be. And so the way I see it, here in the picture is you may start with some inherited mutation, harmful mutation, but essentially through life, we accumulate regularly this replicative or R mutations, to which we can add the environmental ones, of course, can be very important. And so that, to me, made me think, okay, so how can we test this idea in other ways from the 2013 paper? And the idea was very simple. I don't even know if you can really call it modeling, but there is a model. It's extremely basic. It's simply that this lifetime risk, if it's well, let's make the theoretical case where it's in fact only R that causes cancer. Then if it was just that, then the lifetime risk of cancer in a given tissue should be related proportional somehow to the total number of mutations that tissue accumulates in its lifetime. And that number is proportional to this product of D times N, where on one side you have how many divisions you have per cell lineage, times how many stem cells you have in a given tissue. And that, and I'm almost done answering your question, this probably was long, but that was, the 2015 paper where we found this really major correlation between that product, essentially. It's not exactly the product, but it's quite close, and the lifetime risk, across many orders of magnitude. Now, one important thing is that, this result, it's an association, first of all, and did not claim that we were explaining the number of cancer cases. Here is data science department, so everyone is very well aware that this can only address variation in cancer risk. But still, Bert always told me, if epidemiologists found some type of carcinogen that was associated in this way across orders of magnitude with cancer, it would have been definitely important news, and we think it was.
So this picture solicited quite a bit of controversy. I've heard people who say that this should be in every cancer biology book going forward. I've heard, and then you get the two typical types of reaction to sort of a shift in paradigm. There's a subset of people who say we've known this for 30 years, and there's a subset of people who say this is impossible. And in addition, because these replicative mutations were presented to some extent by you, but then in a way that it was amplified by the press and social media as the bad luck factor, the paper got a lot of press as well as controversy. Looking back at the 2015 and that phase of your both academic and personal life, and do you have any thoughts and any lesson learned about how to tame the monster of the press releases and the related consequences?
So here is I guess three things I learned. And again, I have some slides also for this part. So, you know, we had from well-known statisticians attacking this result, to, you know, Nature articles, to to, you know, the WHO making a press release about 10 days after you know, the paper came out. So my first reaction was I actually, a part of me was quite scared, honestly, because I mean, imagine, you know, you publish a paper which I was pretty excited. I thought was interesting and relevant, but I certainly didn't expect the reaction, and certainly didn't expect WHO to, you know, to release a statement against the paper. So you know, you asked for things I've learned. Some of the things that I learned are, well, one, you know, never ever give an interview, allow, you know, to have an interview with a journalist without agreeing, even at the beginning, right from the beginning, that you will go word by word over that final text before it's published. Okay, so that's my rule number one now. If I, you know, I always ask journalists to do that now. And you still, you know, I have this funny story where later with the second paper, I did this, you know, exactly this. And so we were very careful. I was very happy with the article. And then the article came out, and the title was saying 2/3 of cancer cases explained by bad luck. And so, you know, I emailed her, and she was super nice, and she was more upset than me, but she said, you know, it's the editor that decides the title. And I made very clear that that's wrong, but they wanted to go with that anyway. So yes, there is some part of it that maybe it's unavoidable, but I definitely think I learned better how to deal and control some of it, at least. And you know, and then maybe to answer again your question more fully, I think the most important thing is to answer scientifically to those criticisms, and that takes time. And you know, sometimes you just want to go online and post something on Twitter, and then, you know, you control those feelings, and you wait 3 years, and then finally a paper comes out with some answer. So I'll show you, you know, just very briefly. Well, you're familiar with this one. For example, this Nature paper, what they tried to do with our data, they tried to do exactly what I just said that you couldn't do with our data, which is, you know, try to address a number of cancer cases. So essentially on the absolute scale instead of the relative scale. And so, you know, by picking a lower line, measuring how far, you know, with the tissues that had the lowest risk relatively to their total number of stem cell divisions, see how far each of these tissues was, and that will give you a proportion of the number of cases. And this, of course, it's just, it's not possible. We wrote about it. You were one of the co-authors. You know, we could play the opposite game and do the upper line, and then everything becomes bad luck if you do it in that way. And so, you know, it's just not, these points are too noisy to individually to estimate the contribution at the single point level. And again, and we show that, in fact, even if we started with a perfect relationship, say we were 100% correct, it's all the luck, and we had the right model, which of course we know it's not. So it was a straight line. Once you introduce some noise in the data, and then you approach it with that way, where you take the lower envelope, and now you calculate distances, essentially, no matter what this line is, you're always going to get this, the distribution, this proportion. If you repeat this experiment, you know, 10,000 times, you always get the answer that 90% or so will be environmental. So that was one, and that took a long time to publish. And the other that's dear to me, because I think it's very important, is the criticism by WHO. Mainly, their point was that we use a U.S. population, and if we looked somewhere else, it would have been different. And so that is based on this. That I think it's kind of like the poster boy math right? Of why cancer is environmental. With a huge variation across cancer types. And we have some work that is not published yet, but I think it will be coming by the end of this year, and where we show that map. So the map is the same here on the top of the tree that you see here. It's just different colors. That, you know, if you correct for data quality, which is a score that they provide, it's provided by WHO, then of course there are still important differences, but it's not even close to what you know, the original map is. But and maybe one of my points here is that, you know, this thing should be said, you know, when that map is presented, this should be said, because it becomes, I think part of why the paradigm stay like that and for that long is because this type of elements were not included in the discussion of it. So yeah, we did the analysis. We showed that it actually, you know, the result was, the correlation was essentially the same across all countries. I mean, there was variation, but, you know, within the same confidence interval of the United States. And we provided some estimates too for, and here it was a bit different from the 2015, because here we're actually looking at the proportion of mutations that you can attribute to the different factors. So here I think we made one step forward. And you know, we can talk for a long time, so I'll stop here. But I guess the most important thing of this part is not so much the estimate, because it's based on what we know today. And so many have said this could be an overestimate, and it may be. If you ask me, actually, I think it's the opposite. I think it's an underestimate, because we were very conservative in how we did it. But the important point of this paper, it's in the supplement, where we provided this bound, and we showed that essentially, even assuming an extremely high effect of the environment, that I think every epidemiologist would agree it's highly improbable to be that high, you will still get at least 40% of the mutations are due to R. So that's I don't know if this answer, but.
Thank you. No, this is fantastic. I want to remind the attendees, as we have a few more who have made it to the session, that we have a Q&A box, and you're welcome to pop in questions at any time. So, you know, I'll try to keep an eye on what appears in there and try to work them into the conversation, or maybe resurrect them at the end, depending on how things evolve. But you're very, very welcome to add your questions to the Q&A session, particularly as we visit the section of Cristian's Odyssey that are particularly controversial, and also we are covering details at lightning speed as well. So please feel free to slow us down if you'd like. In the meantime, I'd like to maybe move on to some of the collaborations that you since had with some of the folks here at Harvard and otherwise, in trying to reconcile and build a sort of a piece going forward between the novel perspective that you brought to the fore and the cancer prevention community. The cancer prevention community found itself in a difficult position as a result of your papers, because you undermine some messages which, in my estimation, as well as that of most of us who are trying to move that ball, were helpful in trying to get people to adopt behavior that could ultimately result in reduction of cancer, like, you know, smoke less, drink less alcohol, eat more fiber, etc., etc. And it's a tough fight, and having a paper that even puts, you know, the thought in the back of someone's mind that it's a lot of it is bad luck would undermine that work, and as a result, you ended up on the blacklist of many really well-meaning individuals who are fighting the fight of cancer prevention and cancer early detection. So what's impressive about what you did is you've been able to build bridges to this community, engage some of the leaders, and come up with joint work and statements that essentially identify a common scientific consensus that can be used to go forward with both legitimate, rigorous modeling and well-meant policy-driven cancer prevention. Can you tell us a little bit more about those efforts?
Yes, thank you, Giovanni. I would say, it was definitely not just me and Bert. It was, I think, all sides wanted to understand this better, and I think there were good intentions on every side. So it was really a common effort. Here is one example that to me was very important, because just a year before, I had a forum with Dr. Willett discussing, debating these two sides. Sorry, and so it was nice to essentially overall agree that that all of this, if you want, three components are important in cancer, and each one has their role. And now the point is not whether it's 2/3 or 50% or even 40%. The point is to understand that they all play some important role that we need to consider when thinking about how to reduce cancer mortality. Similarly, I would say the field, since our paper in 2013, actually, in that particular direction of how many mutations are normal and so on, has made tremendous progress. And especially some groups in England have published a very many papers, and some from the broad too with Getty-Getz. I think by now it's very clear, right, that our tissues are in fact normally full of mutations, and not just passenger mutations, but in fact drivers and clones and so on. And so that was it was nice to see that happening. And as well as it was nice to see WHO including in the 2020 World Cancer Report, a section where they expressed their opinion, but and I'm not saying that we still fully agree, but that's not important. What is important is that now this is an element in the overall picture. And so because in my title I mentioned prevention, just the one bit about prevention I want to mention is that my personal opinion is that since the war on cancer started, by far, I think no one will argue with this, that by far the focus has been on therapies and some on primary prevention. I think primary prevention has been underfunded too. So I'm on the same side. I think epidemiologists are, but I think that realizing that there's this important role for, call it bad luck or R, implies that secondary prevention, so early detection, has really a fundamental role in reducing, to play if we want to reduce cancer mortality. And so I think that if this was not say discovered, we may have been for another 30 years in the same situation where early detection, because of say some conflicting results with PSA and some other, it's still controversial today, as you know better than me. So this, I'm not saying that we know how to do it yet. I think I'm excited. I think we made a lot of progress, but at least as a direction, I think this research point to it as a very important research direction.
There are a couple of questions in the chat that are very pertinent, and I want to address them before we move on. But I also want to stress the fact that you might have found the ideal receptor in birth when you moved to Hopkins, in part because Bert Vogelstein and his lab were the first to sequence the DNA of human cancer in breast and colorectal cancer in the mid 2000s, and I was privileged to be working with them on those projects. And before they did that, we only knew about the frequency of mutations for genes that we could target. We could go after P53, we could go after PIK3CA. They went for the first unbiased, and the hope was to find a relatively short list of drivers that we could target with drugs and open the way to targeted therapy and really significantly accelerate the rate at which we would be able to treat cancer once it occurred. Now, what emerged from those studies was a mess. There were a ton of genes that were mutated, and your work has demonstrated that on the one side, many of those were mutated as a result of natural variation in tissue. But another side of the same coin is that many of those genes do play a role, at least in some cancer for some individuals. And so, the list of potential targets that we would have to drug suddenly looked impossible to tame. And that, in Bert's mind, shifted his focus towards early detection and prevention, even before your 2013 contact with him about the mutation rates in normal tissue. So that is another piece of evidence that makes what you say in this slide particularly germane and important. Finally, even here at the Farber, well, I shouldn't say even, but here at the Farber as well, we have quite an accent now on the importance of early detection. With that editorial note, I want to move to some of the questions from the attendees. The first, at least in chronological order, is what would be the final conclusion about which epidemiologic and genetics factor come together to better understand cancer prevention?
Well, we don't have the final answer. I think the only conclusion I can make is that on top of what was already known, and so the list, you mentioned already some, smoking, UV, and alcohol and lifestyle, obesity, and so on, and BRCA mutations. Now there is this important component to keep in mind. I have some estimates. Again, we don't have to agree or believe them, but I think the conclusion is that, in my mind, the conclusion is that there is an important component that's, causing mutations to accumulate in our bodies, no matter what. And we have today a much better sense of that process than we had 10 years ago. And I think that's progress.
The next question is really interesting as well. What about prevention of recurrences? Have you given thought about that? And do you think the balance of environmental, replicative, and genetics factor is similar to the pre-cancer world?
Well, yeah, that's a great question. And I haven't actually worked in that direction, but definitely it's something very important to investigate, because recurrence is one of the major issues, of course, recurrence to therapy. So if I had to make a guess, I think that, again, much of that process is still has still a major component that's stochastic. It's just random and it's normal.
There are a couple of more, and I'd like to read them to you. Did you see the questions, Cristian?
No, sorry, because
no. Okay, that's all right. Then I'll read them to you. This is from Mirazul Islam. Hi, Cristian. Nice to see your provocative talk. Did you think of modeling environmental factors like radiation and spontaneously generated cancer, maybe in mouse models, to estimate how much different factors contribute in addition to stem cell division rates?
Yeah, so that's a great question. Let me answer in this way. So I'll skip ahead. I just show you one thing about future research that it's in the workings here. So I am actually collaborating with a person. This is a project I initiated because I was really trying to do what this person just mentioned, because I think in a sense you can make those experiments, right? It's expensive. You have to wait for years. We have been waiting for almost 2 years now, and we are very close to provide something that I think will come out toward the end of this year. But one of the things we do there is, we compare just normal wild type mice, so where essentially it's only potentially bad luck if everything else is under control, versus mice that are exposed to radiation. So, an environmental factor, the contribution of an environmental factor, and how much of that, what a difference does that make? And for now, it's radiation just because it's very easy to control, right? You know how much you're giving to each mouse. You know exactly when you're doing that to each mouse. And so here in this slide, actually, what we are going to show is something that we'll see if it holds with the final numbers. So maybe I shouldn't even show this, but we are still a month away to having all the, a month and a half actually away from having complete data. But it's very interesting because in the field of radiation, there is this thing called fractionation, which is that if you fragment a certain dose of radiation in smaller parts, the assumption is that it's going to cause less cancer. And the intuition is because, if for example, I give it over a period of a few weeks, the same dose, the body is going to be able to digest it better, right? If instead I hit them at once with a very large dose, the body just gets overwhelmed. And that's a biological effect that's certainly true. And there are many experiments showing that. On the other side, what's never been investigated is fractionation in a matter of weeks. But what about fractionation over years? And the modeling that that performing in my lab was saying that actually it should be the opposite in that case. Okay, so there is a biological effect, short-term effect, where fractionation is better if you split it in small doses. On the other side, there is another effect that when you spread it over years, actually you end up with more cancer. And if that holds, that is going to revolutionize a little bit the radiation industry and all the regulations about it. Because now, currently, every time you calculate how much the radiation worker is exposed to over the lifetime to radiation, it's based on this assumption and on this short-term experiments, and the effects could be quite different. Anyway, this was maybe, again, a long answer to the question, but yes, I love that question. That's exactly the type of things I love to do.
This is also a great question for NASA, and I hope that your paper comes out soon enough, because they're trying to scratch their head about how to measure the cancer-related risk of radiation for very long-term missions in space. But without digressing on that, I want to bring up a question from Simona Cristea. She asks, what is your view on tumor initiation? Do you believe it is a particular driver hit that marks initiation, or rather that it is a continuous process? How can we differentiate exponential expansion of driver clones in normal tissue, say skin or esophagus, from exponential expansion in early cancerous lesions? That's a tough one.
So this is a very tough, these are multiple questions, by the way. I'll provide one answer. Let me go to this, maybe. Let's see. Okay. So in 2020, we published, this is a simulation model, because it's complicated enough that we had to do simulations. We are actually now getting ready to provide the analytical solutions for this model. But so what we did here is sorry, let me show. Yeah, here. What we did here is we consider different, we split drivers types in 3 groups, and we consider the dynamics of each one of them from a mathematical perspective, from a mechanistic perspective, assuming 3 different types of effects for those 3 groups, okay? So while cell survival is affecting how frequently division or death rates are, cell fate, it's about the probability of a symmetric versus asymmetric division, and genome maintenance is instead about the mutation rate being increased, okay? And so we did this as it's written there, for the whole, all the phases of a tissue, so from conception to death. And at the cell level, every cell makes these decisions, whether to divide or it dies, and if it divides, how it divides, and which driver, and so on. And this is about 400 million stem cells. So in colon, for example, it's 5,000 divisions each, and we did it across 10,000 individuals. We crushed the cluster a few times. And importantly, there was also a carrying capacity in this model. So one thing so this model provided, sorry, it's a little bit of a long answer, but I think hopefully it will provide an insight. So, we initially feed it by using estimates available in the literature, but we feed it, to colon. We had one tuning parameter basically. So we feed it to colon. And so this is the colon curve, which as you know, it's, you can always do that. And we were not interested in feeding it perfectly, as you can see. But then we did something that I think was interesting, which was, okay, now we don't touch anything. Okay, we feed it, it seems to be working. And this is assuming just the bad luck component. So we are using just the background mutation rate. And then we say, okay, now we switched essentially 4 parameters. And those are, the number of cells, the division rate, those are things that we cannot really choose. They are given to us from the literature. And this model, could replicate qualitatively leukemia and pancreatic cancer, or similarly, say if I increase the mutation rate in Lynch patients, Lynch syndrome patients, it's about 10 times higher the mutation rate. Again, we could get a decent fit to their incidence curve. So this was interesting. And so then one thing that this allowed us to do was to ask, for example, what is the first driver in colon? And what is the first driver in pancreas, right? And I invite the person who asked the question to look at that paper, because what we show is that just because of the way these different drivers work, and this is of course only one piece of the answer, but like it was, you observe a lot of KRAS being first driver in colon, but never reaching cancer, or essentially almost never, right? It had to be of the type, the APC type. And in pancreas was the opposite. And the idea behind is that pancreas divides very slowly normally. And so if you don't hit him first with a driver that acts on the division rate, you're not going to get ever to cancer. Instead, in colorectal cancer, you have a tissue that divides very fast. So actually a KRAS that increases that by a little bit doesn't make a major difference. Okay, but this was the results of the simulation model. It was the output of that. So I think that addresses, at least in part, that type of question. I think it's very important for us to study, understand better and better what effects different drivers have. And of course, the answer is that it's tissue specific. But yes, that's a very—
Thank you, Cristian. Thank you. I really like that paper, and in part because it has, I think a reincarnation of Simona's question is, what can you tell us about the waiting time between the occurrence of driver mutation in a cancer population? It's just the other side of the same coin. And in typical Tomasetti style, your answer is very counterintuitive to me, and probably will get me to rethink about how I'm, whether or not I'm able to put myself in the shoes of a cancer cell or not. But if you are a cancer, so I'll give you my naive intuition, which hopefully you'll debunk live for me. If you are a cancer cell, wouldn't you want early on to to acquire a driver mutation that increases your mutation rate and therefore decreases the time you have to wait until the next driver happens. And if so, these results, which compare the T1, T2, T3 waiting time and show that the time increased from the acquisition of you have to wait longer between driver 2 and driver 3 than you wait between driver 1 and driver 2, right? Am I reading this right? So that would go counter to the intuition that I just provided. Why am I wrong?
I don't think it's counter. I think that what you were saying, the first part is it's exactly what this picture shows, which is that you know, in the previous typical picture, because the assumption was exponential growth, unlimited exponential growth essentially, you can wait a long time because then these things are going to go very fast, very quickly. Okay, and so you can wait 50 years for the first hit. Well, it turns out that once you actually simulate the model and you include all phases and you say, you know what, we have logistic growth actually. You cannot go beyond a certain size unless there is a driver fitness increase and so on. That completely changes the picture, turns it around. So now the first hit has to be, as in general, at around 10, 15 years. So it's exactly what you were saying. Like the first hit has to be quite soon. Now, I can show you in the simulations that if I just don't condition on cancer, so here the part that's maybe counterintuitive to the average reader is the conditioning, right? If I don't condition, then of course, the more I wait, the more I get and I see. But if I condition getting cancer, the first it has to come very early. Yeah, so that's the basic. And you know, here in this case, it shows that it increases. In Lynch, it's about flat. It's probably possible to have something that increases or it's uniform. The case that we couldn't reproduce in general was exactly the typical one from the past. Yeah. This acceleration was for very weird parameter values, I would say.
Thank you. That's terrific. So let me take a look. Oh, so we have maybe 10 more minutes or so, and I was hoping to touch on a recent paper, of which this is one, and maybe if you would like to maybe put this paper in a little bit more perspective rather than presenting it as an answer to some questions that came up, because it's a very, very comprehensive mathematical model of tumorigenesis, and you might want to give us your big picture. And then maybe we'll move on to your work on signatures before wrapping things up.
Sure. Let me maybe, okay, so for this paper, the one thing that, so I think one key was, this was actually a figure in one of Vogelstein's reviews from 2013, the famous science review that he wrote. And one day after probably the 100th times I looked at this figure, it just hit me. And I thought that, of course, if we want to model it properly, we have to understand what is the difference mechanistically among those. So this was kind of like the origin of this paper. But one thing I wanted to say, because it's very dear to me, and I'm sorry, we'll talk about. So let me just mention this because it's, as I said, to me, this is a very important point. So we did it with simulations, and the model, maybe the only thing I should add to that is there are lots of other insights that I invite people, if they are interested, to take a look at the paper. I think it's a very rich paper with information that probably many just discarded immediately, right? Because there is a major part of the field in cancer research that if it says just, mathematical model, okay, this is like, this is something. And I respect, I understand, but so I invite those that are interested to look at it more. But one thing that to me was important there was that one of the criticisms had been that bad luck and the background mutation on its own could not explain cancer incidence across different cancer types, which is exactly what this paper shows, that in fact it's actually relatively easy thing to do. But the other, of course, is that it provides a lot of insights that, even in terms of screening, it provides information on early detection, for example. One of the major problems for those that do work in early detection, developing, say, algorithms for that, they will know, right? One of the problems with cell-free DNA and blood is that you have a lot of clonal expansions, in normal expansions in the blood. So now you want to understand those expansions, you want to model them, understand how big they can be, you know, what is the game, what is the background? So this, I think, hopefully in a good direction. And so I wanted to make a, you know, we were tired to crash the cluster, and these are really big simulations. So I wanted to see if we could make it into an actual, you know, closed form solution type of model. And we had to make some approximations, but I think we kept the key ingredients. And this is going to be submitted in a month, my plan. So it's still in preparation. But a few things I wanted to show about this, I think, are exciting. One is that in this paper, we're going to show that, in fact, with these approximations, when you look at these timings of the drivers, it ends up that you can use the Weibull. Okay, so now the Weibull has been used statistically already for decades, you know, in this type of settings. But I think it's very nice that now we can show that a mechanistic model of tumor evolution ends up being very well approximated by this distribution. And so, you know, now you can still use the Weibull as you did before, but now there is a foundation that's more solid, in my opinion, than what was before, you know, previously. And of course, you can, you know, now having the expressions, you can predict how many mutations you find in a patient versus how many you are actually observing when you do sequencing. So, you know, how many would you predict just based on bad luck, or bad luck plus smoking, and how many did you actually observe, and things of this type. But again, the thing that most dear to me is this one. A major issue in 2015 raised by some mathematicians was the slope of this correlation was less than 0.5. Now, if you think about it, the slope should be at least 1. Why? Because, well, if you think about number of cells, if I double the size of my organ, and these two are, say, completely independent, then I should have essentially double risk, okay? And so it should grow linearly with slope 1. If I think about the division rate, actually that is a power law effect, so even more than 1. But the slope was in fact lower than 0.5, so why is that? And I wasn't too worried because, you know, there is a lot of, you know, this is the first analysis, and but what this model now is allowing us to do is allowing us to do a better analysis, essentially, where we split in one direction the number of cells, which is n here, right, and in the other the division rate, d, capital D, and see what you get there. And this is kind of cryptic, but once you introduce the fact that different tissues have different number of drivers, now you get slopes and estimates that fit much better with the picture. I have the statistical values and all of that, but, you know, we don't have time here. But the point is that this figure fits the reality, in my opinion, much better than the 2015 paper with much better slopes. And so, you know, one thing that, in a sense, is from a statistical perspective, the way I think about what I do, is that, you know, I'm very interested in reducing model bias. Okay, I think that is a critical role the modeling can have, which is to push that envelope closer to the truth. I mean, at the end of the day, the truth, you know, what we call reality is just a model, right? It's our model of reality. And so I think that one of the important roles, especially for applied statisticians, is to get that envelope closer to the truth.
Thank you, Cristian. This sounds like a good time and a good place to start about wrapping this session up. Before I do that, I want to open it one more time for Q&A. There are a couple more, so maybe a 1 minute each. First question. Which are probably the factors that could accelerate to get more driver mutations?
Which are the factors that you said?
Yes.
I would say from the senator work, for example, we think inflammation. It's a very clear, factor that increases the division rate. So I would say that would be, on top of this background, inflammation is what we find, in a sense, across the board.
And that's the perfect segue into the next question, which is asked by Ed Giovanni, who's a pioneer of trying to understand the role of inflammation in cancer etiology. He proposes a simple back of the envelope calculation for you to comment on. He says, nice presentation, in the simple model of 3 required mutation and EHR as being independent, and assuming 60% are R mutation, is it true that for a tumor to be fully R, entirely determined by replicative error, the odds would be 20% of tumor are completely R? That is 0.6 times 0.6 times 0.6.
Yes. So let me answer in this way. I think the most important point to answer this question is that, say, even if out of 100 mutations that are related to a cancer, okay, it's not 100. We know that probably typically drivers are maybe 2 or 3, but let's say whatever the number is, so say 100, even if 99% were due to bad luck or R, but there was one that was caused by the environment, right? That cancer will be still 100% attributable to the environment, as well as 100% attributable to R, because you take away any of the 2 and you don't have cancer, right? So it's a little bit, and that's why I like to focus on proportions of mutations, because it's very hard to tell, right, of a single individual case, how much is just one factor, how much is another. And so yes, so if you the problem with that assumption is that I don't believe in the independence on all of that, but let's say, the idea that if 60%, so the fact that on average is 60% means that, in my opinion, there are many cancers, for example, like smoking, which takes a big share, because in those estimates, it's incidence-based, the estimate. So lung cancer takes a big share of that. And in lung cancer, it's more than 2/3 is due to smoking. So that forces many other cancer types that are more rare or relatively rare than, lung cancer is 10%, 12% versus, some that are 0.1%. Forces many other cancers to have a very large fractions due to R. But again, one driver mutation caused by an environment is still enough to make it preventable. So I don't think we are still there to be able to, really say, based on these calculations, what is really preventable or not. And that's why in our paper we used Harvard's data, the best epidemiological studies we could find, and we trusted those studies.
Thank you, Cristian. This is perfect timing. 1:59:43 just gives me a couple of seconds to thank you from the bottom of my heart for doing this. I hope, and I'm very confident that you will have inspired many of the attendees to take on mathematical modeling as one of the really, really important weapons that we have in our fight for cancer prevention. With that, thanks to everyone for attending, and the ball goes back to our master of ceremony, Erica, for whatever closing rituals we may need to abide to.
No closing rituals. Thank you both for participating today.
Thank you, Erica. Thank you, Giovanni. Thank you so much. Thank you, everyone.